Okay, so welcome to this next video in which we're discussing the hallmarks of cancer as laid out by Hanahan and Weinberg in their 2000 review. Right, okay, so far we've seen that the acquisition of oncogenes, the inhibition of tumor-suppressed genes, the replicative immortality, and angiogenesis. Now, number 5 then is resisting apoptosis. Resisting apoptosis. So basically, not committing suicide, having mutations that mean that the apoptosis pathways are less likely to occur. Okay, so examples of how you could get mutations that lead to being able to resist apoptosis. A archetypal example would be a gain of function mutation in one of the genes encoding for the BCL2 protein. Now, the BCL2 protein is what's known as an anti-apoptotic BCL2 protein, basically. So it binds to the proteins BAX and BAC, which are the pro-apoptotic BH123 proteins. So let me just draw a picture of mitochondria. And let me remind you of the way that the intrinsic apoptosis pathway happens. So if this is our mitochondrion, here are cristae of the inner mitochondrial membrane. And like that. Then basically, in the outer membrane of the mitochondrion, there are proteins known as BH123 proteins, and they are pro-apoptotic. So an example is BAC. Okay. Now another pro-apoptotic BH123 protein is BAX, which isn't in the outer membrane of the mitochondrion, but is instead in the cytoplasm. Okay, so these BAC proteins and these BAX proteins, what they can do is they can aggregate into BH123 aggregates, so pro-apoptotic BH123 aggregates, like so. So you can get the aggregation of BAC and BAX proteins into these BH123 aggregates. Okay, so this is a BH, remember, stands for BCL2 homology domain, so BH123 aggregate here. Okay, right. And these BH123 aggregates basically function as a channel which allows cytochrome C to leave the intermembrane space of the mitochondrion, and then cytochrome C is then going to cause the cell to commit apoptosis when it goes up in the cytoplasm, because usually it should be exclusively sequestered to the intermembrane space of the mitochondria. Okay, so how does BCL2 work? Well, basically, BCL2 binds to these BH123 proteins. For instance, it binds to BAX, and it will bind also to BAC here, and basically it stops them from being able to commit suicide. So let's have it in this turquoise colour. So this was going to represent our BCL2, and here is our BCL2 protein here. So basically it's bound to this BAX protein here, and it's bound to this BAC protein here, and basically it stops this aggregation, so you can't form these BH123 aggregates, therefore you can't let cytochrome C out, therefore you can't commit suicide. So if you get a gain of function mutation in BCL2, then you're obviously going to have increased binding of BCL2 to BAX and BAC, and basically it's going to be harder to drive the cell to actually commit apoptosis via the intrinsic apoptotic pathway. So a gain of function mutation in BCL2 can lead to resistance against apoptosis, basically, and it can stop the cell from committing apoptosis. And if you stop the cell from committing apoptosis, then you're obviously going to divide more. You're going to have more cells, more survival, basically, and that's one of the hallmarks of cancer. Okay, so that's number 5, and now finally, number 6 then, is the one that's very, very important, because this is the one that distinguishes cancer from benign tumours, and it's that it needs to be invasive and metastatic. Okay, so it needs to be actually destroying healthy tissue. because there's a difference between, this is really important, this is the difference between a benign tumour and a malignant tumour. So a benign tumour is basically just loads of cells over dividing. So it's cell, 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 loads of these cells which are dividing too much and have formed a mass. But the mass is not destroying the healthy tissue. Now, benign tumours aren't necessarily harmless, it has to be said. If you get a benign tumour in, let's say, your esophagus, then it may well obstruct the esophagus, and it could press on the trachea, stop you from breathing, and, you know, benign tumours can cause problems. They if they obstruct important tubes, then they can be very, very deadly. But they are not invading healthy tissue and destroying it. Contrast that to a malignant tumour, which is what cancer is. Cancer is a malignant tumour, which is why often some people will refer to malignancies when they mean cancer. So a malignancy, some people will refer to. And that basically means cancer. So cancer or malignancy or whatever you want to call it, malignant tumour, these are actually invading and destroying healthy tissue. And when they invade and destroy healthy tissue, so let's, for example, draw the example in the gut. Okay, let's say we've got the colon here. Let's say here is our tumour cell invading basically the lumen of the gut. Well, sorry, not the lumen, the wall of the gut. And then if this is a blood vessel down here, what they might do is invade onto the blood vessel, and then they might throw cancerous cells into the bloodstream, basically. And then what can happen is these cancerous cells can get lodged elsewhere in the body, and then they can set up secondary tumours there. So that's the concept of metastasis, that a primary tumour can then lead to secondary tumours elsewhere in the body. And it's linked to invasion because you need to invade the blood vessels in order for cancerous cells to get into the bloodstream. Okay, so a very important feature of cancer then is that the cells have this ability to invade, destroy healthy tissue, and then to invade into the bloodstream and then metastasize and form secondary tumours in other sites of the body from where the primary tumour formed.